Hello and welcome to an episode of Betterbee's Interview with Experts series. And I have the great privilege of being here with my scientific colleague on this trip that I have taken up to NOD, the manufacturing facility for Formic Pro, everyone's favorite formic acid-based varroa miticide. So this is Dr. Heather Broccard-Bell, who is going to be sitting here with us for a little while talking about her background and her ongoing research and what we know and don't yet know as much as we'd like to about Formic Pro. So hello, welcome. Can you give us maybe just a quick summary of sort of what your role is here at NOD?
First of all, I'd like to say thank you for having me on this podcast.
Whatever it is.
Whatever it is, yeah. So I am the honeybee health researcher here at NOD, and my role, broadly speaking, is a little bit of research and development. I oversee any field trials that happen here with the bees. We also have a new and growing education center, so I'm involved with that as well. And we do things like hold public speaking events, of which David is going to be one speaker at, actually tomorrow. And on top of that, I travel around and talk to beekeepers and scientists around the world about formic acid and Formic Pro in particular. I also do things like write articles for things. I take pictures. It's a pretty varied job. So combining like science and education, broadly speaking.
Which is really what I do. So it's nice to have a colleague in whatever weird role each of us has carved out for ourselves. It's nice to see that both of our companies are recognizing the value in whatever we're doing and then allowing us to do that. What's interesting is that we actually have a strangely overlapped or sort of overlapped beekeeping background, which is that we both are academic scientists and we both worked in the same academic lineage. So my doctoral advisor, Tom Seeley, was the doctoral advisor for your postdoctoral advisor, James Nye. So in some way I'm not quite your academic uncle, but I'm your academic step-uncle, great-uncle? What's a postdoc? I don't know the rules.
I don't know.
Yeah. But your path to beekeeping did not, for many years, did not seem like it was going towards being a professional bee scientist. So can you just sort of talk people through your varied background that got you to this point?
Yeah, sure. So I think broadly, I grew up in rural Western Canada in Saskatchewan, and I was always interested in animals, and in particular at that time, horses. Although we always had cats and dogs and other kinds of animals growing up.
But no bees.
But no bees. Though I will say that my dad's dad, my grandpa, was a beekeeper. But I never went out and saw his bees.
You never interacted with those bees.
I never interacted with the bees, but he would always give us honey every year.
So you knew that beekeeping was a profitable endeavor.
Yeah. And in Saskatchewan, where my grandpa was a beekeeper, of course, it's full of canola. And so it was canola honey all the time. And that's kind of my experience with honey growing up is canola honey. And now I live in the East where it's a totally different...
Oh, cool.
Yeah. Okay, so I was interested in animals. I was interested in horses. I thought that I would be a veterinarian. I went to university for a year to study pre-vet med. I wasn't probably ready for university. I decided that I would actually pursue horse training professionally for a little while, so I did. I went and I worked in the horse industry, primarily with cutting horses. These are like cowboy horses, until I was 25, and then I decided, I'd like to study animals maybe at a more scientific level. I went back to Saskatchewan, so I had been living in Alberta, which is the province next to it. Went back to Saskatchewan, decided to enroll in university. I did a psychology degree. So that's kind of, at that university, they didn't really have an animal behavior program. So that was the closest that I could really get was doing psychology. So I did a psychology degree. From there, I became interested in neuroscience. I decided that I would pursue neuroscience, which is kind of the biological aspects of psychology. Right. What is the machinery that makes this all happen? Yeah. There was an excellent program in the province over in Alberta, in Lethbridge, doing neuroscience. I decided that I wanted to study play behavior and neuroscience. So there was this excellent program that I knew about but I also looked up to see who was studying play behavior, and it turned out that the one guy in Canada, the main guy who was studying animal play behavior, was at Lethbridge. So
Great.
yeah, I applied
That's
there.
handy.
Yeah, and it was great. It was super, super great. So I took a trip over and met him and his wife, and they were fantastic. He ended up being both my master's and Ph.D. supervisor. Kind of transitioned from looking at developmental neuroscience and play behavior in rats, which is what I did for my master's, to more in a broad-ranging exploration of behavior in general for my PhD. And I was interested in how animals interact with one another. And I'd always been looking at two-way interactions, so like rats fighting with each other or
one animal and another animal.
That's right. And I thought, well, wouldn't it be interesting to look at something more complex than this?
3 animals.
Yeah.
3,000 animals.
Exactly.
30,000 animals.
So actually, initially I thought that I would look at ants because ants are a nice system to work in.
Sure.
And in the meantime, I had decided that I would move down to California. I was trying to find a position, a postdoc position in California. And I talked to a guy who was studying ants, and I wanted to do some computer modeling. And he said, well, I'm not really into modeling, but there's this other guy that works in the department, James Nye, who studies bees. Does that sound like that would be interesting?
And you were like, bees are pretty close to ants. I guess they're close enough that I can tolerate them.
Hymenopterans, right? So
yeah.
fine. So I went and talked to the bee guy, and like that, the rest is history. That was 2013 when I set that up. I started the postdoc position in 2014, and that's really what got me interested in bees. And initially with James, I was studying communication. So I was actually interested in how they're interacting and how they're sharing information and the networks that form.
I'm a bee with knowledge, you're a bee who doesn't have it. How do I pass that to you, and then who do you pass it to?
Exactly. And what happens when I change how that's occurring? And there's many parallels between what's happening in a beehive and what's happening inside your brain. So the way that brain cells share information has many parallels.
I'm a cell and I know something, but you don't.
That's right.
So I'll pass it along and
the kind of the structures and how that happens, very similar. Yeah.
Wow.
Yeah. So that's how I got into bees, and then from there, I was studying this very to what most beekeepers would think of very obscure science. It was just a matter of practicality. We had some trouble getting funding for that kind of research. So James had some other funding that was funding more applied research. So I started doing research on Nosema and pesticides. So I also was doing work in that area. And then from there, decided to move back to Canada. And it was lucky for me.
The stars aligned.
Exactly. There was a job posting, and they were looking for.
They wanted a bee health scientist who would be willing to move to Canada.
Yeah, and
like,
somebody who could also drive a tractor. And I was like, well, with my days with the horses, I could also do that.
I can do all of it.
Yeah,
Terrific. That's fantastic.
Yeah. So that was a long and rambling story.
No, it was fantastic. I think it's important for people to understand when you stand up at an EAS conference or write an article for ABJ or something, for them to say, oh yeah, I understand Heather and her background, and for some of us, we're boring. We just go and we study bees and then we have a PhD in bees. But you've got all of this interesting, diverse background, which I think the more diverse any scientist's training and research has been, in many ways, the more interesting or the more creatively we can think about the current problem we're working on. So I think that it gives you an opportunity to veer off in interesting and exciting directions that maybe you won't expect, and neither will anyone else. That's great. So let's talk about Formic Pro. That is at least a large part of why we're here.
Yes, indeed.
If it wasn't for Formic Pro, a lot of people wouldn't have any bees, so they wouldn't need either of us to teach them more about beekeeping. So I guess I have a couple of questions that I want to get into that I think some of our viewers might be interested in too. And one of them, which I often am asked, and I'd love to get your take on it, is what do we know and what do we not know about, or what do we think we know about the actual mechanism through which formic acid vapor kills a varroa mite?
Yeah, so that's a really interesting question. So it turns out that we actually have quite a lot of research on the effects of formic acid in animals, actually in organisms in general. It's been fairly widely studied. It's a molecule that's been under study for at least a couple hundred years. We first started studying it in ants, which is how it got its name.
Right.
That's the
Formic,
Latin,
right,
Latin word for ant. And it turns out that it's the simplest organic molecule. It is everywhere. It's a part of biochemistry. There's a ton of things happening in your body and my body based on formic acid.
So I've got formic acid in me right now.
That's right. It's being produced as a byproduct of many chemical processes. It's also an input into the many chemical processes.
And if there's too much of it, there's processes that are supposed to clean it up and get rid
That's
of
right.
it.
Like it's bad if there's too much, but it's also
bad
bad
if
if there's not enough. Yeah.
Right.
So your body like tightly regulates it. So we have quite a lot of information studying what formic acid does in the bodies of lots of different organisms. And it seems like a fairly universal finding that what happens is if you have too much formic acid, it actually interferes with your body's cells' ability to produce energy. So cells need energy to function. Without energy to function, cells die. If all your cells die or too many of them die, then you also die. So if you interfere with that process, it causes cell death.
Right.
So formic acid, when the level is too high, interferes with that process. So we know that from a bunch of different animals. We actually don't directly know that from studying it in Varroa, but it's wide enough.
It does kill them, and we know why it kills other animals, so we can make an educated guess.
Make an educated guess, and this is a system. This biochemical pathway, how cells produce energy, is what we call evolutionarily conserved. So highly evolutionarily conserved. Basically, everything that's alive.
Everybody does it the same way.
Uses, or not exactly the same,
way.
but this pathway is essentially in everything we've ever looked at.
And it's also in honeybees.
And it's
so
also
why
in
doesn't
honeybees.
it kill all the honeybees?
That's right. So the other part of this is, yes, it interferes with the cell's ability to make energy, but the body has ways of contending with the levels that are too high. And one of the ways of contending with it is by producing what we call detoxification enzymes. So these other molecules that are produced by the body break down formic acid when there's too much of it. And we know now directly from research in Varroa and honeybees that when you apply formic acid to a honeybee colony and you look at what's happening inside the bodies of those organisms, the detoxification enzymes that are being produced by bees and the ones being produced by Varroa are slightly different, and there's different amounts of them. So it's what seems to be the case, now it's not definitive, but what seems to be the case is that honeybees are simply better equipped to deal with excess levels of formic acid than Varroa.
Which makes sense.
It kind of makes sense.
Right? You know, a honeybee has to go out anywhere into the world to all sorts of things. A Varroa mite gets to live in a nice climate-controlled environment like a bee colony, and so maybe they depend upon the bees to keep things safe and clean and not have all of these toxic chemicals floating around.
Yeah.
So now they're less equipped to deal with it if you squirt a bunch of it into the hive.
That's right. Like, yeah, what you're saying, like the Varroa's food source, which is the bees, is very consistent. Whereas a bee is going out and foraging and gathering different things.
Right.
Formic acid is also produced as a byproduct of plant biochemistry, so it's in all of the plant material.
Right. It's naturally found in honey because it's naturally found in the nectar.
Absolutely.
Right.
But there are varying levels of it out in the environment, whereas the bees produce a fairly consistent level. So the Varroa don't have to contend with excess levels of it as much as the bees will.
Until we soak it into a pad and then put it into the colony.
Yes,
And
unless
they weren't
we
expecting
really
that,
overwhelm
were they?
them. Yeah, yeah.
Okay, well that's fantastic, thank you. So Varroa mites aren't the only mites that people are concerned about. There's also Tropolaelaps mites, which are found in Southeast Asia, and we've got increasing evidence that they've been spreading or at least have been found with us looking more diligently in India and parts of Central Asia and Eastern Europe. So do we have any reason to think that Tropolaelaps mites could also be controlled using formic acid?
We do, actually. So people have been, well, first of all, in the places where Tropolaelaps has been known to exist for quite some time, people have been beekeeping with it for a while, and they've actually been using liquid formic acid to control it. So we do know that it's effective. And there have been a couple of groups who have tried our product, Formic Pro, to control Tropolaelaps mites, and it seems to work quite well. And so we're actually running currently in the process of setting up a trial to do a formalized test of that.
Right, to do a real rigorous scientific study of Formic Pro for Tropolaelaps control.
Yes.
But we have evidence already that is making us optimistic about that. So
I would be very surprised if it came up like this totally doesn't work. Like I don't think that will.
Which is handy, right? In the same way that formic acid molecules are quite good at killing Varroa, unsurprisingly, they would also be good at killing Tropolaelaps.
Yeah. Yeah.
Fantastic.
You know, an animal that's not that distantly related.
Right.
So, yeah.
So working in Thailand sounds pretty fun. Are you going to get to go to Thailand to do that work?
Oh, I'm really hoping so. Yes, yes. I'm wanting to be involved in a few aspects of the actual trial. So yes.
Terrific. Yeah. And so I guess Thailand also has another added wrinkle to it, which is that it is a place that is often quite hot. And I know that when I talk to beekeepers, and I rave about how wonderful Formic Pro performs in Betterbee's apiaries, I do sometimes get some pushback from folks in warmer climates. They say, it's too hot for me to use Formic. I can't use the Formic, and so I'm not going to be able to do that. Do you feel like there are opportunities for people to use Formic Pro effectively in warmer climates, or do you think that it's just not really compatible with really hot places?
The question is always, for how long is it hot? So I think one real misconception of Formic Pro and how to best use it is that you need to ensure that you have the entire duration of the treatment window be under a certain temperature, within a certain temperature range. And this is actually incorrect. So if you actually look at the label, it says...
It's a 2-week treatment period.
That's right. But you don't need 2 weeks, you know,
below 85 degrees Fahrenheit.
And because people write and they're like, well, but how do you even know? Like, how can I plan this far in advance?
Yeah, right. The weatherman only gives me a 10-day forecast,
I don't know
and I can't
with
even
any
trust
certainty.
that. Right.
But what it comes down to is really a lot of that formic acid has dissipated within the first 3 days.
Okay.
So as long as you're within that temperature range in the first 3 days, then you are fine. And most places, at least some time of the year, there will be a window where you have 3 days.
It's just a cooler period of 3 days long is sufficient for you to go
below
and use
85 degrees Fahrenheit or 29.5 degrees Celsius.
Got it. Yeah. Got it. Okay. And so that still sort of raises the question, though, if I'm in a place like Thailand or Florida where I don't have that 3-day window, could there be other ways that Formic Pro could be used, or that formic acid could get formulated, that you think would be effective in a very different climate? Or does it just not work that way?
Okay, so first I'll tell you what the issue is currently. So the people think that it's very straightforward, and it's not. It turns out that it's not actually that straightforward of a problem. Above around the beginning of brood rearing temperature, which is right around the upper range for where you apply formic acid, it's a little bit higher than what the upper range technically is. At that temperature, the behavior of the bees changes, because for them, they need to maintain that brood at a certain temperature for it to develop properly, right? So above that range, they start to fan a lot. They need to cool it down. What happens when you have a miticide that is a vapor and you're trying to maintain a certain concentration in a hive?
But I've just turned on, 10,000 fans.
Exactly. So what happens then, if you've got a big, bustin' strong colony, you're going to flush out so much formic acid that it might not actually work that well. And that's really counterintuitive, because people think that the problem is that the temperature is too hot.
Too much formic.
And so it's evaporating too fast.
Yeah.
But that's not really the case, or could be the case, but not necessarily. Again, big bustin' colony, you have too much fanning, and you actually end up underdosing them. On the other hand, if you've got a colony that's weaker, and you've got formic acid in there, and because they're really only tightly regulating directly where the brood is, they may not be as tightly regulating where the strip is if it's not close enough to the brood, or if there isn't very much brood in the colony.
Okay.
So the strip might be subject to closer to ambient temperatures, which would make it evaporate faster.
Okay.
And now you've got a small colony.
You need the strip to be in that climate-controlled brood zone that the bees are regulating, but you can't have the bees all sitting there fanning their wings desperately to cool it down.
100%.
Okay, so it's really, the formic is engineered to be in that sort of predictable, consistent brood nest.
Ideal circumstances, you're going to get as close to the brood as possible for multiple reasons. One is that. The other one is that this is a brood treatment. Like, we're trying to get one of the big benefits of using formic acid is that it's able to penetrate the brood cap. The closer you are to where that brood is, the better it's going to be able to do that. Right.
Okay. And this is the same reason, to frame the high temperature issues that way as just not having that consistently regulated brood nest that it was engineered for. The same is true for why we can't use formic acid when a colony is in cluster in the middle of the winter.
That's
is
right.
that they don't have that ventilation and that activity going on, so it's just not going to behave
So
appropriately.
in the wintertime, you know, you've got your Formic Pro strip in there, and the cluster is much smaller, the brood is much smaller, so they're really not
If there's
regulating.
any at all.
That's right.
Right.
They're really not going to be regulating most likely where the strip is directly. So you're going to have a way slower release. But then add to that the fact that the colony is not very active, so they're not going to be fanning it. So you actually could. I wouldn't be able to accurately predict, is this going to
overdose or underdose.
I don't know.
Right,
Something
right.
bad is going to happen.
Yeah.
That's what it,
Don't
that's
do
why
that.
it's, that's
Nobody
what I say
do
to
that.
people. Yeah. At the end of the day, what it comes down to is when you're applying it outside the temperature range, the results become unpredictable.
Got it.
Yeah.
Got it. So it's not even that it won't kill mites or it will kill bees. It's that you can't reliably and confidently say what will happen.
That's right.
Which is the issue.
Right.
But that really, fundamentally, for most beekeepers who are worried about these weather limitations, it's I think that understanding that it's that 3-day window where it needs to be below the label temperature, and that the rest of the treatment period is just letting things work themselves out after the treatment has begun. But that main formic what would you call it, sort of pulse, is coming out just in those first few days.
And that's not to say that there isn't any formic acid being released after that, because there is. I mean, we have lots of data looking at.
There's still more.
That's right. And it is still high enough to kill mites, but you're sort of out of the danger zone in terms
your
of
sort of major
overdosing.
spike is right in those first bits.
Yeah. Yeah. So that's not to say, though, that there might not be ways that we could modify how it's applied or, you know, do something to engineer it to, and we are working on those things.
Great. Good thing NOD has a scientist here working on things like that, right? You know, you don't want to have a product and think, well, it could be better, but they're not going to put in the time and the effort and the money to get someone to make it better. So,
but I will say it takes a long
here.
time to do these things.
Right.
So
right. Science doesn't happen when you snap your fingers.
Yeah.
I guess on the same subject, you and I share a sort of a similar role in that we talk to a lot of beekeepers who are thinking about using Formic Pro, or they have used Formic Pro, and you know, things have gone well, or things have gone poorly, or things have confused them, and now they want us to clarify. Do you, in your expert sort of position here, talking to beekeepers like that, do you feel like there are particular common misconceptions that you want to, you know, set the record straight on? We've already talked about that temperature limit, which I think is an important one, but are there other sort of common mistakes that you want to prevent people from making?
I would say that the 3 biggest considerations when you're thinking about using formic acid, Formic Pro in particular, but this also applies to other versions.
Right.
Number 1, appropriate temperature. So we've already covered that. Number 2, this is a vapor. A lot of the other miticides that people use are contact chemicals, so that
the bees have to touch it, and then they have to
right.
touch each other, and then they have to touch the mites.
And in that way, it doesn't matter what size or shape the cavity is. It works the same as long as the bees are contacting it.
Because the vapor will just expand to fill.
But
or you're saying that
I'm saying for the contact,
works.
it doesn't matter.
But this is a.
But a vapor will, yes, it will expand to fill the cavity.
But you've engineered this for particular cavities.
That's right. So we've done a ton of testing on cavities of certain sizes or within ranges of sizes. So we know exactly what the right dose is, so that we can target Varroa without killing bees.
Right.
So to get back to like how
the toxicity. It's much less toxic to bees than to mites, but enough of it will still kill bees.
Absolutely.
So you've got to be careful.
So I do like to say, like, it's a medicine like any other medicine.
Right.
You know, we've dialed in the dosage to target what we want, but it's also toxic too.
Right. If I can take enough antibiotics that I die along with the bacteria, I just shouldn't.
That's right.
Right.
Yes. So the size and the shape of the cavity actually matter. So that's actually a benefit of formic acid is that it's a vapor and it expands to fill the cavity.
Right. It'll get to every corner, but you've engineered it for particular shapes.
Yeah. So we don't have to rely on the bees wanting to touch it or not. They're going to get touched by it. It's going to get to the mites. That's great. That's a benefit. But then we have to
right.
recognize.
But if I have a long horizontal Langstroth colony with a weird modified entrance, and I've changed this,
it may not work the same, and we don't know.
Yeah.
You know, as we expand into areas where they have different kinds and sizes and shapes of hives, we do more testing on different sizes and shapes. We can only do so much, and to a large extent, beekeepers are very, very ingenious people, and they're often modifying their equipment. So we can only tell you what to do in the most common hive styles, and we might not necessarily know what to do if you have something that we haven't seen before.
So temperature, remember that it's a vapor.
Remember that it's a vapor, so the size and the shape of your cavity, and especially the ventilation of your cavity.
Right. Don't leave your entrance reducer into your Langstroth hive when you put your Formic Pro strips in.
And pay attention to the fact that formic acid is heavier than air, so it kind of doesn't matter what's happening at the top. People ask us about upper entrances all the time. It doesn't matter because it's not really going out there. It's the bottom entrance that really matters. You want to make sure that entrance is all the way open if it's a Langstroth hive, but
If I have a screen bottom board.
that's right. So we do want to maintain a certain concentration of formic acid, otherwise it's not going to work very well. So you want to make sure that your screen bottom board has the tray inserted. You want to make sure that that's...
So basically it's engineered to be producing this heavy formic acid vapor that's dropping down, and then the colony is supposed to be losing that vapor at exactly one entrance's speed, right? You know, that volume.
It's not quite that precise.
Well, sure, because you can use an 8-frame or a 10-frame hive and they'll both
There's
be okay.
a little bit of... But this is like we've done 20 years of development on this to try to dial this in as well as we possibly can. The third aspect that's really important to consider, so like I said, there were three, is the size of the colony. So if your colony is too small, they are not going to be able to effectively move the vapors around enough to flush out enough, and you really...
So they could get overwhelmed.
Overdosing it.
Okay.
Yeah. So too small is bad. Big is fine. You may notice, however, if you put it on a big busting colony, that they'll come out and they'll beard on the front. And that's fine. That's absolutely fine.
Because the goal is to be treating the mites in the brood, and the brood aren't going to be wandering out into that beard. They can't move. So you'll kill them even if you've got one mite clinging to one bee who says, I don't want to be in here, I'm just going to stay on the front porch all night. You know, there's going to be a couple of mites that make it through one way or the other. So that's a tolerable...
No viroicide is 100% effective. It's just not possible to develop something like that.
If there was, we'd all use it for one day, and then we'd never have to think about this again.
That's right. We'd never have to... But like, if anyone can name a disease like that, or like any sort of a pathogen like that.
Okay, terrific. Yeah, that's really helpful. So I guess to that thought about big colonies and small colonies, one thing that people often don't remember or maybe don't fully understand about Formic Pro is that unlike most of these miticides, there's two different ways to apply it to your colonies. The label that gives the official legal instructions in the United States for how it can or can't be used says you can apply 2 strips, and then that's a 2-week treatment period. Or you can do 1 strip and then wait a period of...
It's 10 days.
10 days, and then put in the second strip, and then you wait another 10 days or 7 or so.
So the whole treatment period on the what we call 1+1 method is 20 days.
Okay, but the 1+1 or the 2-strip treatment are both effective and approved treatments, but why might I choose one or the other? Does that have something to do with colony size or temperature? Or when might you lean one way or the other? Or maybe we don't quite know which of these would be the better choice.
What I will say to that is I personally never use the 1-strip method.
You use the 2-strip?
I use the 2-strip method regardless. If you want to make sure that you're really getting at the mites under the capped brood, use the 2-strip method.
That's the most reliable way to really make sure you're penetrating all of that brood. And just don't use it if the colony's quite small and may get overwhelmed.
Yeah, exactly. Like
just
read
use
the
another
label.
miticide.
Read
or
the label.
Wait a little while until the bees grow.
Yeah.
Okay, got it.
Yeah. And actually, I should add a little addendum to what I said before about the conditions under which you should apply Formic Pro. Another fairly important thing is to make sure that your colony is not stressed. So if you have just split them, or you've just come out of winter, or you've just come out of pollination, you've just moved them, wait for them to settle in, or wait for them to grow to sufficient size. Formic acid, like any other miticide, is a stressful treatment.
Right.
So you don't want to add stresses to stresses.
So is that because the individual bees might be more harmed by it, or is it because the colony, when it's stressed, isn't really sort of structured and prepared for the extra challenge of ventilating this new chemical that's been put in?
I'd say it's probably a combination of both.
Okay.
So individually, it depends on what the stressor is too. Sure. So for instance, you're just coming out of winter and your bees are nutritionally stressed. So individually, I think individual bees would be affected by that. But it also affects the ability of the colony to adapt to any perturbation, any change in the environment, such as formic acid.
Got it. Okay. I guess one thing that I want to do before we wrap up is I do want to give you a chance as a scientist to talk a little bit about your science. So do you want to talk about some of the things that you've been able to do, some of the things you're currently doing, some of what you might have in the future? Obviously, you know, maybe there's some things you can't talk about, but anything that you can talk about, because I know that in addition to doing research on, you know, how to make better products or new products or whatever else you might do, you are given free rein to do research which is simply of benefit to beekeepers. You're allowed to do experiments that will just help us keep our bees healthy and thriving.
Yeah, so I'm really lucky to work at NOD, where we have you know, a lot of the company staff, including the CEO in particular, are very, very supportive of basic science. And so
well, your CEO is a beekeeper, so any research you do that helps his beekeeping, he's going to be happy with.
That's right. So we've been fortunate enough to we were able to get some funding, and I had a collaborator in North Dakota where we were able to run a 2-year study looking at viruses, and we were focusing on deformed wing virus. We were looking at A, B, and C, and we were tracking how it moved through colonies, and not just how it moves through colonies, but how it moves through populations within colonies. So we were looking at adult workers, adult drones, and then pupa of drones and workers, and ones that were infected by Varroa and ones that were not parasitized by Varroa. So because we know that DWV is transmitted by Varroa, but there's some evidence that there might be some differential transmission among those strains.
Of different strains.
That's right. So how does it move? And then we also looked at what happens if we apply a miticide to those levels. Now we're still finishing analyzing all those data, but I'm hoping to write up a publication for that. So you know, it's Formic Pro obviously was involved in that, but the main hypothesis, we were actually asking the question about the role of drones in that whole system. So, because drones, we know that Varroa preferentially parasitize drones, adults and pupa. So it makes sense that at certain times of the year, if they're transmitting a lot of virus, that's where the virus is going to be
sort of circulating in the drone bodies more than some others.
Exactly. And then at other times of the year, which is the fall in our climate,
there are no drones allowed.
There's no drones. So what happens, the Varroa has to go somewhere. So it's going to go into its non-preferred host, which is the workers.
Got it.
What happens to all that viral load? Does it get transferred?
Fascinating.
And I will say that the findings that we had were not as straightforward as what we had predicted, but that's the exciting thing about science.
Right. If we knew what the answer would be, we wouldn't have to ask the question in the first place.
That's right. Yeah,
why would we get so hot and sweaty out there doing the data collection if we knew exactly what it was going to tell
100%. So the other exciting project that we've been working on for the last couple of years is, I mentioned that we have been trying to adapt Formic Pro use to different hive styles. So in Europe, and in particular in Spain, there is a style of hive called the Layens hive. It's a little bit like a top bar hive. I think probably more people are familiar with that. Top
bar-ish.
The frames fit together tightly at the top. So whereas in a Langstroth hive, you can lay Formic Pro on top of it, and it works just fine because the vapors can go down through the frames.
You've basically got a wall in a Layens hive.
You can't do that. You can't do it. So as we started selling Formic Pro in Europe, and especially in Spain, we had to figure out how or if it was possible. So we've spent 2 years. We have 36 custom designed Layens hives for research here. We actually ended up doing 3 full studies, 3 full field trials, where we figured out that we can actually hang the strips in between the frames we tried different orientations and in different locations. I was really surprised because what we ended up finding out is that the best place to do it is to hang it right in the heart of the brood zone and like squish 2 frames together.
This is a vertical Formic Pro strip hung right down in between.
We had these like plastic mesh bags. When it goes to becoming a formal method of application, we'll have like something standardized to put it in. But for prototypes, we had these plastic mesh bags.
As scientists, we never get to use the fancy stuff.
Exactly.
We use
it
like to use.
Nail something together and it's fine.
It's us with push pins hoping that it'll stick. Right.
So we have we have these vertical strips of Formic Pro, and they're hanging between 2 frames. You push the frames back together, and I really thought like this is going to be a massacre. Like there's just going to be a bunch of dead brood. It's not going to work. And it turns out, and we've replicated it twice, that it does a way better job at controlling the Varroa when it's in the brood zone. And on top of that, when we actually asked the question like, how much brood is in each of these groups that we treated,
Right.
there's no difference. So
there's just as much brood in the colonies
That's right.
that had it slammed right down in the middle of the
even though
brood
you can
frames.
clearly see on the brood frames that where the strip actually was, like there's a
those
hole there.
those brood didn't do great,
those
but
didn't.
the brood in general
Exactly. In
were
general,
just fine.
there's just no difference. So
That's great.
Yeah.
Fantastic.
Yeah.
It's so exciting to see someone doing work that genuinely answers beekeeper questions. I have always valued my ability to be able to reach out to you as a colleague and say, hey, I have a customer, they've put in Formic Pro, they've observed this interesting behavior, they've got this question that I wasn't able to answer, and that we're able to talk about it, and that sometimes even those questions then cause you to say, well, you know, I'll do that experiment next summer. We can run a field trial. We can do a test. We can talk to some of our other colleagues and see what we can put together. So it's really valuable, I think, for you know, it's valuable for me to have you here at NOD, and I know that NOD's whole philosophy really aligns very well with Betterbee's. That, you know, at Betterbee, our mindset is that we want to be beekeepers serving beekeepers. We, you know, they've hired a scientist, and one of my jobs is to make sure that everything we do is scientifically valid and validated, and that I test and question things that maybe haven't been investigated. But it really, I feel a great deal of alignment with what we try to be, with what NOD tries to be. Can you comment a little bit on sort of why it is that you guys do things the way you do them, or what your goals are? Are you an evil company like all companies to trying to make money?
We're not.
Right. but what is it that you guys think this is all about?
I mean, I have to say I'm really proud to work for NOD, because NOD does have a really strong business ethic, and the ethic is focused on, yes, we're beekeepers, you know, designing products for beekeepers. We recently computed, I don't remember what the exact number is, but it's something around 150 years of beekeeping experience at NOD. Among
you
all
take everybody's
the
years of
staff.
experience
That's
and you stack it up, and that's great.
Our CEO is a former, actually kind of current as well, commercial beekeeper. So he, you know, and he's the guy who started the company. We have, I think, 6 people who are former or current commercial level beekeepers at the company. So it's quite a lot. So that, of course, beekeepers designing things for beekeepers that will be useful for beekeepers. But then on top of that, we also really strive to produce products that are sustainable. So things that, and I mean sustainable in the sense that, you know, we're doing minimal damage to the planet, but also things that are sustainable in that we might be able to use them for a long period of time. So we talked about the mechanism of action of formic acid, and people say, you know, people think that it's very unlikely that Varroa would be able to develop resistance to it. I agree with that, and one of the reasons that I agree with that is because it is such a fundamental part of biochemistry. Formic acid is everywhere. It would be difficult to, I mean, no organism has yet figured out how to deal with life without using formic acid.
Varroa have to have some formic acid in their body at the right concentration. So if they developed something that totally purged it, they'd kill themselves.
Yeah.
And so it's hard to fight. It'd be like if we tried to fight, you know, having sugar in our blood. You know, you need blood sugar, you're not able to do anything.
So using something like, using that as an active ingredient where it's everywhere, I think that's a different mindset and a different approach, and allows us the possibility that this will really be a long-term sustainable product.
Right. That my great-great-grandkid beekeepers might very well be using Formic Pro.
And we've been using formic acid for 40 years.
3.0 or whatever it might be.
40 years, and there's not a hint that it doesn't work anymore.
Excellent.
So,
and especially in the light of recent research that continues to show that some mites are continuing to develop more resistance to more of the miticides that have been on the market. To know that there isn't any evidence in that realm for Formic Pro is comforting for somebody who's going to use it, and also for us to think in general about our beekeeping. Is if we can shift beekeeping into a way that can use more of these more sustainable treatments, maybe everybody's lives get easier.
Yeah. And that goes back to our you know, our company mandates and missions. You know, NOD stands for Nature's Own Design. So we're using an active ingredient that's already out there in nature, which is formic acid.
Right.
And
formic acid is naturally found in bee venom. It's naturally found in honey. It's a molecule that's sort of supposed to be in there.
Yeah.
It's just coming in at a slightly higher dose during the treatment period.
That's right.
But nature has already found this to be useful to the bees. You're just giving it a supercharge.
Absolutely. Yeah.
Yeah, that's great. Well, Heather, thank you so much. It has been a great pleasure talking to you. I hope that anybody who has any further questions, you can reach out to me at Betterbee. You can reach out to Heather at NOD, because we are always happy to help you be better beekeepers. And so it's been our great pleasure to sit here and chat with you for a bit, and look forward to the next interview with whoever else I can trick into sitting in front of a camera with me for a half hour.
Thanks, David. It's been fun.
Absolutely.